Hey guys, in this lesson I'm going to show you how to do a Punnett square for a monohybrid cross. Okay, so we're looking at example 2 from the PowerPoint, and the problem says Mendel self-fertilized 2 pea plants from the F1 generation in the previous example. In other words, both parents were heterozygous. Use a Punnett square to show the genotypic and phenotypic ratios. So the first thing we want to do here is figure out what our alleles represent. And in this case, the dominant gene, Y, is going to be equal to yellow. And the recessive gene, lowercase y, is going to be equal to green. After we've determined that, we're going to write the genotypes of the cross. So I have a big Y, little y, which is heterozygous for one parent, in black, and I have big Y, little y, which is also heterozygous for the other parent. Why are they heterozygous? It's because you have different alleles. You have a dominant allele paired with a recessive allele. These genotypes can then make sex cells. The alleles are going to segregate during meiosis, and one sex cell will have the dominant gene, and the other sex cell will have the recessive gene. And the same happens for this parent. And here you can see I put the little tail on to indicate that they're sperm. So what we want to do next is draw a Punnett square, and in the Punnett square we want to show all the combinations that these sperm can make with these eggs. So how do we do that? So we're going to place the letters that represent the genes in these sex cells for one parent on the left side of the Punnett square, like that. Okay? And then we're going to put the other parent's sex cells on top, just like that. Then we're going to complete the Punnett square by writing the genotypes inside each of these boxes. So what we're doing is we're combining sex cells here. These two sex cells are going to combine, and we end up with big Y, big Y. I use different colors again to represent that each parent is contributing their copy of the gene. So in this box we have big Y, little y. Notice how I wrote the big dominant gene first. And here we have big Y, little y. Again, always write the dominant gene first when you're completing the Punnett square. And in this one we have little y, little y. So from this we can figure out what our results are going to be by calculating the genotypic and phenotypic ratios. So, for example, there's 3 genotypes that we're going to get from this cross. The first one is big Y, and that occurs 1 out of 4, or 25% of the time. So I'm looking at this one right here, 1 out of 4, 25%. The other genotype is heterozygous, big Y, little y, and it occurs 50% because there's 2 out of 4 boxes that have that genotype. And then lastly, we have little y, and that genotype occurs 1 out of 4, or 25%. Okay? Remember, genotypes are the combination of alleles. It's the letters that we're looking at there. With phenotype, we're looking at the observable characteristic that results from a particular genotype. So in this cross, what we're going to have as a result is really 3 out of 4 are going to end up being yellow. This one's yellow because it has both dominant genes present. This one is yellow, and this one is yellow. What do all 3 have in common? They all have the dominant Y gene, which covers up or masks the recessive. Lastly, we have the green phenotype, which is going to occur 1 out of 4, or 25%, and that will be the one that I color green. And here, the only possible way to get a green phenotype is to have 2 recessive alleles matched up. Okay, So that's how you solve a genetics problem. I hope that was helpful, and we'll see you next time. Bye.